Hi, and welcome to Pharmacology of the Synapse. In this video, we'll review the mechanisms of G protein receptor action using the opioid receptors as an example. GPCRs are called GPCRs because they couple to a G protein complex composed of alpha, beta, and gamma subunits. The cycle of GPCR activation starts with a signaling molecule binding to the receptor. This can be a ligand or agonist for the receptor. For example, morphine or beta-endorphin would bind to the mu-opioid receptor, triggering a signaling cascade. Next, the alpha subunit of the GPCR exchanges GDP for GTP, which prompts the dissociation of the alpha subunit from the beta-gamma subunit. These subunits can go on to trigger a variety of signaling responses. We'll talk about the different kinds of responses and how the responses are generated in the next slides. The last step of the signaling cycle is the hydrolysis of GTP back to GDP, which is assisted by a regulator of G protein activity, or RGS protein, and results in reassociation of the alpha and the beta gamma subunits and resets the system for further activation. The alpha and the beta gamma components can result in the activation of many different signaling cascades, so resulting in many different kinds of responses such as ion channel opening or closing and the activation of second messenger cascades. So how do GPCRs cause such a wide range of effects? GPCRs can have a wide range of effects due to three different mechanisms: multiple downstream effectors for each GPCR, multiple coupled G proteins for each GPCR, and the fact that the receptors can form dimers. We will go over multiple downstream effectors in the next few slides. GPCRs have multiple downstream effectors, or molecules or signaling mechanisms, that cause an effect. If we take the mu-opioid receptor as an example, morphine is an agonist at this receptor. And activation of the mu-opioid receptors by morphine leads to activation of potassium channels, inward rectifying potassium channels, as well as inhibition of calcium channel activity and inhibition of cyclic AMP, as well as the activation of the MAPK second messenger cascade. So morphine itself, due to the activation of multiple GPCR effector pathways, inhibits the neuron's ability to transmit pain signals using multiple mechanisms by hyperpolarizing the neuron via its actions on the potassium channels, reducing the activation of the neuron by reducing calcium activity, and by inhibiting cyclic AMP activity. Since the GPCRs have multiple signaling pathways, one pathway can be more activated than other pathways. This is called signaling bias. Some ligands preferentially activate one pathway over another, which is a concept called biased agonism. Biased agonism is very important in drug development, and it's an important concept because it helps design better drugs. If one pathway leads to undesirable effects, such as an increase in side effects, minimizing activation of this pathway would be important. On the other hand, if one pathway is better at producing analgesia, preferentially activating that pathway would be desirable. So for example, one of the signaling pathways after activation of the mu-opioid receptors is the recruitment of beta-arrestin, which is associated with more side effects. So the development of new opioid receptor drugs should minimize beta-arrestin recruitment as much as possible. Next, we'll talk about how GPCRs can be coupled to multiple G proteins, resulting in multiple effects. There are many different types of G proteins, and we now know that a GPCR is not necessarily coupled to just one G protein. These graphs are called webs of G protein activation, with the different types of G protein on the outside and the red line shows how strongly activated the different G proteins are. The further towards the edge of the circle, the stronger the activation. On the left here, you will see the activation profile of the mu-opioid receptor by endomorphin 1. The mu-opioid receptor is usually described as a receptor that couples to GI-GO proteins. You can see that activation of this receptor by endomorphin 1 results in activation of several GI proteins, and also several GO proteins, and also GZ. Activation of the kappa opioid receptor on the right by dynorphin A shows a slightly different activation profile. These G protein subtypes can couple to different downstream effectors, providing the GPCR with another way to produce a wide variety of effects. Lastly, we will go over GPCR dimers, which also results in activation of different downstream effectors compared with single receptors. GPCRs can form dimers, either with themselves or with other GPCRs. In the case of the mu-opioid receptor, it can form a homodimer, which is essentially two mu-opioid receptors, or heterodimers of a mu-opioid receptor with a delta opioid receptor. In terms of signaling pathways, the heterodimers are interesting because mu-delta receptors have a different activation profile compared with mu receptors, such that they activate GZ more instead of GI. Mu-opioid receptors can even form dimers with non-opioid receptors. For example, the mu-opioid receptor has been found to dimerize with the cannabinoid receptor CB1. This heterodimer has been shown to have weaker activation of downstream signaling pathways compared to each receptor alone. After receptor activation, GPCRs can be recycled or degraded. For the mu-opioid receptors, after the receptor and the downstream effectors have been activated, the receptor is phosphorylated by GPCR kinases, or GRKs. Phosphorylation prompts the recruitment of beta-arrestin to the receptor and then causes the receptor to be internalized in clathrin-coated pits. At this point, the receptor can be degraded or recycled back to the surface. The fate of the receptor, either degradation or recycling, depends on the type of receptor, the ligand that binds to the receptor, and the interaction between intracellular signaling pathways. As shown in this video, GPCRs come with many accessory proteins that regulate receptor function. To review, we have encountered RGS proteins, which assist in the hydrolysis of GTP to GDP, G-protein coupled receptor kinases, or GRKs, which phosphorylate the GPCR after it's been activated, and beta-arrestins, which are recruited to a phosphorylated receptor and causes receptor internalization.